9.4 Signaling in single-celled organisms. By the end of this section, you will be able to do the following. Describe how single-celled yeasts use cell signaling to communicate with one another. Relate the role of quorum sensing to the ability of some bacteria to form biofilms within cell signaling allows bacteria to respond to environmental cues, such as nutrient levels. Some single-celled organisms also release molecules to signal to each other. Signaling in yeasts are eukaryotes, fungi, and the components and processes found in yeast signals are similar to those of cell surface receptor signals in multicellular organisms. Budding yeasts, figure 9.16, are able to participate in a process that is similar to sexual reproduction that entails two haploid cells, cells with one-half the normal number of chromosomes, combining to form a diploid cell, a cell with two sets of each chromosome, which is what normal body cells contain. In order to find another haploid yeast cell that is prepared to mate, budding yeasts secrete a signaling molecule called mating factor. When mating factor binds to cell surface receptors in other yeast cells that are nearby, they stop their normal growth cycles and initiate a cell signaling cascade that includes protein kinases and GTP binding proteins that are similar to G proteins. 9.4 Signaling in single-celled organisms. 243 Figure 9.16 Budding Saccharomyces cerevisiae yeast cells can communicate by releasing a signaling molecule called mating factor. In this micrograph, they are visualized using differential interference contrast microscopy, a light microscopy technique that enhances the contrast of the sample. Signaling in bacteria enables bacteria to monitor extracellular conditions, ensure that there are sufficient amounts of nutrients, and ensure that hazardous situations are avoided. There are circumstances, however, when bacteria communicate with each other. The first evidence of bacterial communication was observed in a bacterium that has a symbiotic relationship with Hawaiian bobtail squid. When the population density of the bacteria reaches a certain level, specific gene expression is initiated, and the bacteria produce bioluminescent proteins that emit light. Because the number of cells present in the environment, cell density, is the determining factor for signaling, bacterial signaling was named quorum sensing. In politics and business, a quorum is the minimum number of members required to be present to vote on an issue. Quorum sensing uses autoinducers as signaling molecules. Autoinducers are signaling molecules secreted by bacteria to communicate with other bacteria of the same kind. The secreted autoinducers can be small, hydrophobic molecules, such as acyl homoserine lactone, AL, or larger peptide-based molecules. Each type of molecule has a different mode of action. When AL enters target bacteria, it binds to transcription factors, which then switch gene expression on or off. When the number of bacteria increases, so does the concentration of the autoinducer, triggering increased expression of certain genes including autoinducers, which results in a self-amplifying cycle, also known as a positive feedback loop, figure 9.17. The peptide autoinducers stimulate more complicated signaling pathways that include bacterial kinases. The changes in bacteria following exposure to autoinducers can be quite extensive. The pathogenic bacterium Pseudomonas aeruginosa has 616 different genes that respond to autoinducers. Some species of bacteria that use quorum sensing form biofilms, complex colonies of bacteria, often containing several species, that exchange chemical signals to coordinate the release of toxins that will attack the host. Bacterial biofilms, figure 9.18, can sometimes be found on medical equipment. When biofilms invade implants such as hip or knee replacements or heart pacemakers, they can cause life-threatening infections. 244 Chapter 9, Cell Communication Access for free at OpenStax.org. Visual connection figure 9.17 Autoinducers are small molecules or proteins produced by bacteria that regulate gene expression. Which of the following statements about quorum sensing is false? A. Autoinducer must bind to receptor to turn on transcription of genes responsible for the production of more autoinducer. B. The receptor stays in the bacterial cell, but the autoinducer diffuses out. C. Autoinducer can only act on a different cell. It cannot act on the cell in which it is made. D. Autoinducer turns on genes that enable the bacteria to form a biofilm. 9.4. Signaling in single-celled organisms. 245 Visual connection figure 9.18 Cell-cell communication enables these, Staphylococcus aureus bacteria to work together to form a biofilm inside a hospital patient's catheter, seen here via scanning electron microscopy. S. aureus is the main cause of hospital-acquired infections. B. Hawaiian bobtail squid have a symbiotic relationship with the bioluminescent bacteria Vibrio fischeri. The luminescence makes it difficult to see the squid from below because it effectively eliminates its shadow. In return for camouflage, the squid provides food for the bacteria. Free-living V. fischeri do not produce luciferase, the enzyme responsible for luminescence, but V. fischeri living in a symbiotic relationship with the squid do. Quorum sensing determines whether the bacteria should produce the luciferase enzyme. Credit A. Modifications of work by CDC, Janice Carr. Credit B. Modifications of work by Cliff 1066, Flickr. What advantage might biofilm production confer on the S. aureus inside the catheter? Research on the details of quorum sensing has led to advances in growing bacteria for industrial purposes. Recent discoveries suggest that it may be possible to exploit bacterial signaling. Pathways to control bacterial growth. This process could replace or supplement antibiotics that are no longer effective in certain situations. Link to Learning. Watch geneticist Bonnie Bassler discuss her discovery. http://openstax.org/l/bacteria_talk of quorum sensing in biofilm bacteria in squid. Evolution connection. Cellular communication in yeasts. The first cellular form of life on our planet likely consisted of single-celled prokaryotic organisms that had limited interaction with each other. While some external signaling occurs between different species of single-celled organisms, the majority of signaling within bacteria and yeasts concerns only other members of the same species. The evolution of cellular communication is an absolute necessity for the development of multicellular organisms, and this innovation is thought to have required approximately 2 billion years to appear in early life forms. Yeasts are single-celled eukaryotes and, therefore, have a nucleus and organelles characteristic of more complex life forms. Comparisons of the genomes of yeasts, nematode worms, fruit flies, and humans illustrate the evolution of increasingly complex signaling systems that allow for the efficient inner workings that keep humans and other complex life forms functioning correctly. Kinases are a major component of cellular communication, and studies of these enzymes illustrate the evolutionary connectivity of different species. Yeasts have 130 types of kinases. More complex organisms such as nematode worms and fruit flies have 454 and 239 kinases, respectively. Of the 130 kinase types in yeast, 97 belong to the 55 subfamilies of kinases that are found in other 246. Chapter 9. Cell communication. Access for free at openstax.org. Eukaryotic organisms. The only obvious deficiency seen in yeasts is the complete absence of tyrosine kinases. It is hypothesized that phosphorylation of tyrosine residues is needed to control the more sophisticated functions of development, differentiation, and cellular communication used in multicellular organisms. Because yeasts contain many of the same classes of signaling proteins as humans, these organisms are ideal for studying signaling cascades. Yeasts multiply quickly and are much simpler organisms than humans or other multicellular animals. Therefore, the signaling cascades are also simpler and easier to study, although they contain similar counterparts to human signaling. Point 2 Link to Learning. Watch this collection of interview clips with biofilm researchers in What Are Bacterial Biofilms? Click to view content, https://openstax.org/l/bacteria_biofilm.